Well, good afternoon, everyone, and welcome to Research Grand Round. And I'm not having a stroke or Bell's palsy, just got out of the dentist chair. So that's dedication. And it really is my pleasure this afternoon, despite the timing of dentistry, to introduce Dr. Jason Hellmann, who is an associate professor of medicine here. And he's part of our Envirome Institute, and he is actually going to speak on dietary dependence of exercise-enhanced inflammation resolution. And we're hoping that we have a lot of our internal medicine residents and cardiology fellows involved to try to capture someone for whose interest would align with this and help us do some translational research. Dr. Hellmann is actually a Louisville Cardinal, not quite a lifer. Why? Because after college, grad school, and Ph.D. training here, he escaped temporarily for a postdoctoral fellowship at the Brigham and Women's Hospital at Harvard Medical School. But we're so glad that he came back and that he's been on our faculty since 2018, 16, absolutely. All right, thank you so much, Dr. Hellmann. We're looking forward to all your comments.
Thank you, Dr. Williams. And thank everyone here and everyone that's online that logged in to hear about a little bit of, I figured I would take this opportunity to introduce some of the research interests in my lab, as well as talk about a recent project that we've undertaken. And so you'll obviously see by the title, the mainstay or the main research interest in my lab is really trying to understand fundamental mechanisms that control inflammatory tone and duration, and in particular, how inflammation is resolved. And so you might be asking yourself, well, we have a lot of agents and pharmacotherapies that we can tap into already to turn off inflammation. So, what really are you trying to learn that's new? So we've noticed since the time of Hippocrates that, to reduce fever and reduce inflammation, simply, chew on the bark of the willow tree. So, what else is there to develop and learn? But since then, we've come quite a far ways with obviously the development of steroids, the non-steroidal anti-inflammatory drugs, as well as I'm sure many of you remember the selective COX-2 inhibitors, and as well the new class of drugs that are, termed the biologics to really dampen the inflammatory response by keying in on specific cytokines. But as we know, with almost anything, there's trade-offs in using these drugs. So with steroids, we know with chronic use, you develop osteoporosis, you impair wound healing, increase the risk of fungal infections. All of this is due to, obviously, the immunosuppressive actions of steroids. Additionally, taking a lot of NSAIDs leads to gastric bleeding, cardiovascular complications, kidney toxicity. We're all well aware of the COX-2 story, where those had to be largely pulled from the market due to their increased thrombosis and risk of heart attack. And this new class of biologics, such as anti-TNF therapies, they also lead to increased infections by dampening the immune responses to any invading pathogens or infections. And so what my lab is really interested in is trying to understand how lipid mediators that are involved in the acute inflammatory response may be operationalized as potential therapeutics to combat chronic inflammation. So we know in an acute inflammatory response, within seconds of minutes of the host being invaded or challenged, there's the formation of edema to help facilitate the recruitment of leukocytes to the site of infection, namely PMN or neutrophils, which are recruited very quickly to the site of infection. This is largely controlled by the presence of prostaglandins and leukotrienes, which are critical for vasodilation to help facilitate leukocyte recruitment to that site, as well as leukotrienes, which are strong chemoattractants for the recruitment of neutrophils. But in kind of capturing and keeping these bugs contained, neutrophils actually undergo apoptosis and start to die and must be cleared effectively by monocyte macrophages that are recruited to this area. And this process is largely controlled now that we're starting to appreciate the involvement of a class of lipid mediators called SPMs, or specialized pro-resolving mediators. namely the lipoxins, resolvins, protectins, and merisins. And so what the SPMs do in general terms, is they actually stimulate the macrophages to consume and remove these invading neutrophils that are undergoing apoptosis. And this is nicely demonstrated here in this rabbit ear wound model, where you can see the quickly trafficking neutrophils to the side of the wound, followed by the more slowly migrating macrophages to help clear away these swarming neutrophils. And so this is a relatively busy slide, but I think it kind of drives home an important couple points regarding the biosynthesis of SPMs. And so we know the SPMs are largely derived from EPA or DHA, which are fish oils, omega-3 fatty acids, through the conversion by a couple of different enzymes, but namely 5-lipoxygenase and 15-lipoxygenase. So as I was mentioning, the SPMs are made up of several different classes or different families of lipid mediators. The resolvin E series are derived from EPA, D series resolvins from DHA, as well as the protectins and merisins through the biological actions of 15 and 12-lipoxygenase through the conversion of DHA. And so to this point, several of the different receptors for, a number of these SPMs have been identified, as well as the actions on different cell types have been identified and published in the literature. Some of the signaling mechanisms as well have been identified, but this remains kind of an active area of investigation, trying to understand the signaling mechanisms by which these lipids actually promote the resolution of inflammation. But one of the common themes to the right-hand side, the pro-resolving functions that you'll quickly notice is, downregulation of inflammatory signaling pathways such as NF-kappa B, with a concomitant increase in phagocytosis. And so these SPMs are not simply anti-inflammatory, and that is, they not simply, reduce leukocyte recruitment to the site of inflammation, but they actually promote resolution of inflammation by enhancing macrophage phagocytosis. And so we think because of these distinct actions of these chemical mediators, that they might have advantageous role therapeutically. And so it was the discovery of these lipids that really changed the way we think about how to treat inflammation. So previously it was thought that inflammation, the chemotactic stimuli, passively just dissipated with time. But really, we're starting to appreciate that to turn off inflammation, there are active cellular events and biochemical pathways that might be, exploited to turn off chronic inflammation in particular. And so I became interested in this pretty early on in my graduate career, because we knew from the human studies data that many of the type 2 diabetics, insulin-resistant individuals, presented at the clinic with elevated levels of the CRP, which is a marker of systemic inflammation, as well as the inflammatory cytokine IL-6 and leukocyte blood counts, all correlated with insulin resistance. And at that time, there was a lot of emerging data in the field that were showing that if we intervene at any level of the inflammatory signaling, we can largely protect these animals against diet-induced insulin resistance. And so we became very interested because we knew that the adipose tissue in particular, as these mice were gaining weight and the adipose tissue was expanding, there was a increased release of free fatty acids into circulation, which then allows it to ectopically store either in skeletal muscle or in the liver, which then had pathophysiological effects. Additionally, there was evidence to support that there was inflammatory mediators being released specifically from the adipose tissue that was driving systemic insulin resistance. And so, all of this data really kind of got our attention and allowed us to kind of, or forced us to really ask the question, and maybe this inflammation in type 2 diabetes is a result of a failure of these SPMs to resolve inflammation. So to test that directly, we utilized DB mice, which are hyperphagic animals. We treated them with 100 nanograms of resolvin D1, one of these SPM molecules that I was describing. We simply gave the animals an IP injection for 8 consecutive days. On day 5, we performed a glucose tolerance test and really didn't see any significant differences. There was really nothing going on that was remarkable. We saw, fasting blood glucose levels weren't significantly different. But interestingly, by day 9, we started to see a significant, albeit, maybe small, but it was statistically significant decrease in the fasting blood glucose levels. And so we were questioning whether maybe we just need to go treat these animals longer. So we repeated the study, and this time performed a glucose tolerance test after 10 days of treating these animals with resolvin D1. And again, at day 10, we saw a decrease in fasting blood glucose. And then this time we actually see an improvement in the glucose tolerance test, as indicated in this lower panel. Additionally, we see an enhancement in the levels of circulating adiponectin at both day 9 and day 17, which is a adipokine that's known to enhance insulin sensitivity. And so, given the literature and the involvement of adipose tissue, we started investigating, what might be happening in these animals treated with resolvin D1. So we performed some basic H&E histology to look at the formation of these crown-like structures, which are known to contain or be sites of leukocyte infiltration to help clear away these dying adipocytes. And hopefully you can appreciate that the resolvin D1 animals, the formation of these crown-like structures was significantly inhibited at both day 9 as well as at day 17 following the resolvin D1 treatment. As well as we see a sustainment or an elevation in the presence of CD301 positive macrophages, which are anti-inflammatory pro-resolving macrophages in these animals treated with resolvin D1, as well as the phosphorylation of AKT, which is a downstream mediator of the insulin signaling pathway. We see that animals treated with insulin and resolvin D1 have a significantly increased phosphorylation of AKT. So all of this collectively suggested to us that treating these animals with resolvin D1 was improving the adipose tissue inflammation, which was leading to a protection and a decrease in the systemic insulin resistance that was happening in these hyperphagic animals. So that was one story. We also, in a similar chronic inflammatory model of atherosclerosis, we saw a similar effect if we treat the animals with resolvin D1. It improved the lesion size, lesion stability. There's a whole host of other disease models now that have shown that if we give back these SPMs, we can improve the pathophysiology in the various different chronic inflammatory models. And again, this is busy on purpose to kind of demonstrate now how far we've come. The top table basically is demonstrating, have implicated that there's a decrease in the production of various different SPMs in all of these preclinical models of chronic inflammation, as well as the receptors seem to be downregulated or seem to be modified in some fashion, which is inhibiting the pro-resolving actions of SPMs. And so we think this might be a general phenomenon that we're seeing in chronic inflammatory models, that the signaling of SPMs or even the production of SPMs is somehow being perturbed. And so, my lab in particular, we're kind of struggling with this a bit, where do we go from here? So we really wanted to know, are there endogenous mechanisms at play that we could tap into to enhance the production of these SPMs, or are we always relegated to just adding them back? Do we have to take a pill? Do we have to give a shot? Are there other mechanisms that might teach us something about how these pathways are being impacted in chronic inflammation? And so we started getting interested in exercise. Exercise is known to have robust anti-inflammatory effects, namely the activation of the HPA or sympathetic adrenal medullary axis, its activity on the adrenal gland leading to upregulation and release of adrenaline or epinephrine, and cortisol has direct actions on circulating monocytes to decrease TNF alpha production. Obviously, there's actions on skeletal muscle and the release of this now termed myokine, IL-6, which is a canonical inflammatory cytokine. However, has been shown to decrease TNF-alpha production and IL-1 receptor antagonists in circulating monocytes. There's also actions on the adipose tissue, downregulation of these pro-inflammatory M1 macrophages, and an increase in the upregulation of anti-inflammatory M2 macrophages. And so there's a lot of literature kind of circulating about the anti-inflammatory effects of repeated exercise. And so we were curious if maybe it's not simply anti-inflammatory, but maybe there's pro-resolving actions of exercise. And so to test this, we relied on our this acute inflammatory model of peritonitis, which is a really nice model that allows us to quickly isolate trafficking leukocytes and quantify them using flow cytometry, and also take the supernatants at the same time and quantify these lipid mediators, so that we know these direct associations. And all of this can be done in relatively quick time, in roughly about a week, we can get an answer. So, and just to remind you, we know early on that the acute inflammatory response is dominated by the presence of leukotrienes and prostaglandins, followed by the upregulation of these SPMs and resolvins during the resolving phase. Additionally, this model is well characterized in the literature, and we can actually quantify the resolution index, which will allow us to assign really a quantitative number to how exercise might be impacting this process. And so to do this, we allowed animals to exercise on a forced treadmill model for 4 weeks with increasing duration at 75% of their max capacity, followed by this peritonitis model that I just described. And we quantified the leukocytes trafficking in the peritoneal cavity. And interestingly, what we found is that exercise animals had a robust infiltration of neutrophils at the peak of inflammation compared to the sedentary controls. However, the removal of these neutrophils was enhanced in exercised animals compared to these sedentary animals. And this led to about a 7-hour enhancement in the resolution, which doesn't sound like a lot, but it actually equates to about a 30% increase in the resolution phase of this acute inflammatory response. Given the role of macrophages in removing these neutrophils, we were surprised to find that the absolute number of macrophages really wasn't different between sedentary and exercised animals. However, we did see elevated levels of resolvin D1 following exercise, as well as the expression of ALOX15 and ALOX5. These are the genes for 5-lipoxygenase and 15-lipoxygenase that I was describing earlier. So with this data in mind, really suggests that there might be an enhancement in phagocytosis of these macrophages. So to test that directly, we isolated peritoneal macrophages from sedentary and exercised animals and fed them FITSI-labeled zymazan in a dish. And after a couple of hours, hopefully you can appreciate the increased uptake of this FITSI-labeled zymazan in the exercised macrophages. Additionally, we found that these macrophages produce more resolvin D1 in response to phagocytosis. And so that really led us, okay, that's like really exciting, but what is the mechanism? And so we went on a little bit of a long fishing expedition, trying to find a potential mechanism that might be playing a role here in simulating SPM production. And so we measured cytokines, really didn't see much going on there. And finally, we stumbled upon looking at the role of epinephrine in this process. And so one of the kind of the first hints that might be involved was using bone marrow derived macrophages in a dish. So what we found there is if we treat these macrophages with increasing amounts of epinephrine, it actually stimulates the production of resolvin D1 in vitro. Additionally, it's a little bit hard to see here, but hopefully you can appreciate epinephrine treatment led to an upregulation in the expression of 15-lipoxygenase, which was inhibited, surprisingly, when the cells were pre-treated with the alpha-1 adrenergic receptor antagonist prazosin. and not by the beta 1 or beta 2 antagonists, this ICI compound or taxolol. So this kind of suggested that alpha 1 is mediating the upregulation of 15-lipoxygenase following epinephrine treatment. And to kind of drive that point home a little bit further, we see that epinephrine enhancement in RBD1 is blocked if we block the alpha 1 receptor. Additionally, if we agonize or if we treat with phenylephrine to stimulate alpha 1 directly, we can enhance macrophage phagocytosis as well as RBD1 production. And if we inhibit 15-lipoxygenase activity using this compound ML351, we can block the alpha 1 mediated effect on macrophage phagocytosis. So we then moved back in vivo. That was all in vitro. So we needed to test this mechanism to see if it was contributing in vivo. And so again, we exercised animals for 4 weeks on that forced treadmill protocol and then induced peritonitis. And again, we see this enhancement in neutrophil recruitment compared to sedentary controls with an enhanced removal in these exercised animals. However, in the animals that underwent exercise and then also received prazosin, this alpha-1 antagonist, we can significantly reduce the exercise-enhanced resolution. And here's the quantification of the leukocytes during this process. And so, additionally, we see a significant inhibition of exercise-induced resolvin D1 when we block alpha-1. And we also see that in vivo phagocytosis assay and the uptake of these latex IgG-coated beads that blocking alpha-1 adrenergic receptor significantly reduces uptake in the peritoneal macrophages. So collectively, what we found in this study is that in a preclinical model, we see that epinephrine release following exercise seems to be important in stimulating resolvin D1 production via stimulation of alpha-1 and the upregulation of 15-lipoxygenase to stimulate macrophage uptake and removal of neutrophils. So all of this was in a peritonitis model, all normal chow. And so we really wanted to know next, is this dietary dependent? Based on our previous findings in high-fat fed and in BBDB animals, was this a dietary dependent effect with exercise? And so to study this, we put animals on a low-fat diet or high-fat diet for 6 weeks. Half the animals were subjected to 4 weeks of exercise while also consuming that high-fat diet or not. And interestingly, I think it's important to note here is that this 6 weeks of high-fat diet did not cause any overt insulin resistance or adipose tissue inflammation. This is really looking at the dietary dependence. We see no, inhibition of the animal's ability to adapt to exercise with low-fat diet. Their adaptation to exercise is the same as low-fat fed animals. The amount of lactate that they generate at fatigue during the exercise capacity test is the same. So all of this is to basically say that this dietary change did not affect the animal's ability to perform exercise, even during an exercise capacity test. We see no significant differences in weight gain. We do see a change in postprandial non-fasting blood glucose levels, but really no significant differences if we fast the animals. We see an enhancement in lean mass with exercise. That's the same between low-fat and high-fat fed animals. We see a decrease in fat mass with exercise. That's similar between low-fat fed and high-fat fed animals, as assessed by DEXA scan, and no differences in glucose tolerance or insulin tolerance tests. So really, the 6 weeks of high fat, it's not causing any overt systemic effects on metabolism. But interestingly, when we look at the expression of ALOX15 or 15-lipoxygenase in the adipose tissue, we find that low-fat fed animals have an upregulation in the adipose tissue following exercise that is inhibited in the animals that consumed high-fat diet. Additionally, we find upregulation of multiple SPMs in the adipose tissue in low-fat fed animals following exercise that's inhibited by exercise, either resolvin D1, the so-called aspirin-triggered resolvin D1, resolvin D4, or aspirin-triggered resolvin D3. all four are being inhibited in animals that consume high-fat diet despite undergoing exercise. Interestingly, we also see upregulation of M2 anti-inflammatory macrophages that express CD301, and this is being inhibited by high-fat diet. So we're really curious to kind of tease out what's going on here. So the first question we asked is, what cell type in the adipose tissue may be responsible for the upregulation of ALOX15? What cell type is responding to exercise? And so we separate the adipocytes out from the stromal vascular fraction. We don't see much going on in the adipocytes, but what we see is that following exercise, ALOX15 is upregulated, specifically in the stromal vascular fraction of the tissue. And so, I mean, that was nice, but the stromal vascular fraction is also made up of multiple cell types. And so what we decided to do next was separate out the F480 positive cells, which is a pan marker for macrophages. And so we looked at F480 positive versus F480 negative cells following magnetic bead sorting. And what we found is that it was the F480 positive fraction that was upregulating the expression of ALOX15, and we don't see much going on really with the F480 negative fraction, all the other cell types that are present in the adipose tissue. And interestingly, this effect on the macrophages in this tissue is again inhibited when the animals consume high-fat diet. So kind of taking a step back here, it seems like with exercise, there's an upregulation of ALOX15 in the macrophages of adipose tissue, and high-fat diet is blocking that effect. And we think that this is important because ALOX15 gives rise to these SPMs to turn off inflammation. So next, so how is high-fat diet doing this? And so we first looked at the adrenal glands, given what we found in the peritonitis model and the involvement of epinephrine, and performed just gross histological analysis of the adrenal glands of these mice, and really nothing really remarkable going on here. But when we perform PCR and we look at the expression of PNMT or phenylethanolamine and methyltransferase, which is involved in the conversion of norepi to epinephrine, we see a significant decrease in the expression with high-fat fed or in high-fat fed animals. And this was also bore out in the immunoblotting, showing that protein expression of PNMT is downregulated. Additionally, urinary levels of epinephrine following exercise in low-fat fed animals goes up, and high-fat diet is blocking that, despite the animals performing the same amount of exercise as the low-fat fed animals. So this was really kind of the first hint that maybe high-fat diet is doing something to the adrenal gland, which is preventing, the release of epinephrine, which then was having what we think are signaling effects on the adipose tissue. And so we're still kind of trying to hunt down a mechanism by which high-fat diet is inhibiting the expression of PNMT. But so far, what we've kind of landed on is that the adrenal cortex and the release of glucocorticoids actually signals through the classic glucocorticoid receptor alpha in the adrenal medulla to control the expression of PNMT. And in the literature, there's a described splice variant of this glucocorticoid receptor called glucocorticoid receptor beta, which lacks the ligand binding domain and will form a heterodimer with GR alpha and actually serve as like an inhibitor to the GR alpha receptor. And what we see is that high-fat feeding leads to increased expression of glucocorticoid receptor beta, specifically in the adrenal gland. So we still have a lot of work there left to do. But this all suggested to us that, maybe high-fat diet is inhibiting the release of epinephrine following exercise. And so we did a simple experiment. We put animals again on the same feeding regimen that we were doing before, 60% high-fat diet for 6 weeks. And this time we didn't do any exercise, but instead we treated animals with epinephrine or vehicle control. And we treated them with low-dose epinephrine during that last week of being on high-fat diet, trying to, replicate, if you will, this effect of exercise. And so what we see in the adipose tissue is that epinephrine treatment leads to recruitment of more macrophages, But surprisingly, they were both M1 phenotype as well as the CD301 positive M2 phenotype. So we able to recapitulate some of the findings that we see with exercise, but a little bit of a different response here. And that epinephrine was enhancing just total macrophage abundance, probably in response to lipolysis. But importantly, we added a third group. These animals were pre-treated with that 15-lipoxygenase inhibitor ML351 before receiving the epinephrine treatment. And what we found is that ML351 did not have any effect on epinephrine-induced recruitment of macrophages to the adipose tissue, but inhibiting 15-lipoxygenase led to a significant increase in the presence of M1s with a decrease in the presence of M2s in the adipose tissue. So what we think is going on is that epinephrine is important for getting macrophages to the adipose tissue, but then 15-lipoxygenase is important for dictating whether those macrophages become M1 or M2. And so we're still trying to kind of work this out, but we really have now focused on how macrophages control the expression of CD301 that we're using to identify these M2s. And so given what we know now in the literature that intermediary metabolism in macrophages is coupled with their phenotype, we looked at how CD301 expression might be changed in response to these changes in intermediary metabolism. So for this, we took bone marrow macrophages and treated them with IL-4 to induce that prototypical M2 phenotype. And as expected, or has been reported, we see an increase in the expression of CD301 with IL-4 treatment. But interestingly, when we add palmitate to the culture media to help facilitate mitochondrial respiration and beta oxidation, we can further drive up the expression of CD301, which is inhibited if we block beta oxidation. So what this is suggesting to us is that CD301 expression that we're using to identify these M2s is directly coupled with fatty acid oxidation in bone marrow macrophages. And so we took a step back and we isolated the stromal vascular fraction, and we treated these cells with resolvin D1 and then used XF seahorse analysis to get a sense of oxidative metabolism by looking at their oxygen consumption rate. And what we find is that in the SVF fraction, these fractions treated with resolvin D1 have a significant increase in basal OCR, suggesting enhanced mitochondrial respiration, as well as an enhancement in their maximal OCR, which is potentiated even in the presence of palmitate. And so we think SPMs are stimulating fatty acid oxidation in macrophages, which is then driving the CD301 expression and M2 phenotype. We also think it's not specific to resolvin D1. We see a similar effect with resolvin D4, and even in the context of adding oleate to the cell culture media and not just palmitate. And so all of this was in preclinical models, and so we're interested, as Dr. Williams alluded to at the beginning, and we're interested in trying to see if these findings translate. So to try to get at a hint whether that might be happening or not, Dan Riggs and a talented student in my lab at the time, Ellie Farley, we started doing an analysis of the NHANES data set, this national nutrition survey that's been ongoing in the US for quite some time. And so using data from 2003-2004 NHANES data set, where they've actually measured circulating levels of fatty acids and have performed VO2 max assessment on a subset of these subjects and looked at C-reactive protein abundance as a marker of systemic inflammation. We were able to identify 435 participants that we could use to really kind of answer whether or not this relationship between exercise and resolution might be occurring in a human. So the first thing that we observed is that VO2 max, as you increase your cardiorespiratory fitness, these individuals have decreased levels of circulating C-reactive protein, which is a marker of systemic inflammation. So this isn't new data. This has been shown in numerous other papers, but it helps to validate the use of this NHANES cohort to kind of get at whether or not exercise might be having some effect on systemic inflammation. But interestingly, when Dan started doing some analysis looking at saturated fatty acids, monounsaturated or polyunsaturated fatty acids, interestingly, what he found is that it was really the levels of omega-3 fatty acids in particular that seem to be important effect modifiers in this relationship between CRP and VO2 max. To say it in a different way, individuals that had elevated levels of omega-3 fatty acids experienced a more negative slope in the relationship between VO2 max and CRP. And in particular, it was the levels of alpha-linoleic acid as well as docosahexaenoic acid, which we know is a precursor to SPMs, that is really important for driving this effect modification. So this isn't, clear evidence that exercise is enhancing SPMs to turn off inflammation, but it's providing some rationale to maybe do more of these studies in the future. And so we're in that vein, we're collaborating with a investigator at NYU on the Perspire trial. This is the pro-resolving and pro-inflammatory response to acute exhaustive exercise in healthy individuals. And the hypothesis here is that acute exhaustive exercise will elicit increases in circulating SPMs that is changed based on training status. So just as a way of a quick summary, we are enrolling participants into the study and then doing blood draws before and after a cardiopulmonary exercise test, waiting 4 hours, collecting, doing a blood draw again, and then subjecting these individuals to 3 months of exercise, lifestyle intervention, and then bringing them back into the office and repeating the same similar kind of blood draws. And then we're going to perform deep immunophenotyping, looking at different immune cell subsets, as well as looking at the levels of SPMs. And so we're really excited. I think we've enrolled maybe 10 participants at this point. So stay tuned for that. But in summary, obesity-induced insulin resistance is associated with delayed resolution of inflammation. And we think promoting resolution improves insulin sensitivity and decreases fasting blood glucose levels, at least in preclinical models. Exercise stimulates the production of SPMs and enhances macrophage phagocytosis via adrenergic stimulation. We have evidence to suggest it's probably alpha-1 mediated. And the pro-resolving response to exercise in adipose tissue, at least, seems to be dietary dependent, as these animals that consume high-fat diet do not see this pro-resolving effect following exercise. And I didn't have time today. I actually went a little fast. I probably did have time. But we also have evidence to suggest that the SPMs are promoting beta oxidation, fatty acid oxidation in macrophages via AMP kinase. And so hopefully, made a compelling story that exercise is not simply anti-inflammatory, but rather pro-resolving. So keep that in mind while you're out running, maybe not in 90-degree heat, but so. But with that, I'd like to acknowledge Ernesto Pina-Calderon. It's been a very talented grad student in my lab who's done a lot of this work, as well as Jenny, who's been in the lab since 2016. She's really the mainstay, kind of the go-to person in the lab, as well as Nolan Boyd, who does a lot of our mass spectrometry data acquisition and analysis. And as I mentioned, Dan for his help with the ongoing studies looking at how SPMs may differ in different populations, as well as some of our collaborators intramurally and extramurally. So, and with that, obviously, our grant support. So with that, I'll take any questions if there are any.
Pretty open for questions. The one that pops up is the high fat food model that you were using.
Right.
Which kind of oils? Because I was going to give you a whole thing, but you did it already about there are oils that are pro-inflammatory,
right,
those that are not.
Yeah. So this is a 60% high fat, and it's basically lard. It's a lot of saturated fat.
Absolutely. Okay.
Yeah.
So And so that makes perfect sense.
Right.
And so you could kind of repeat it with, olive oil, so anything
Right.
like monounsaturated fat.
Yeah, and see if the effects are replicated or not.
Right, or magically the M2 macrophages will wake up.
Right. Yeah. Sanjay?
So very, very nice talk, Jason.
Thank you.
You were talking about not running in 90 degrees, right? So that's related with that. What do we know about this resolution of inflammation, especially your SPMs? They're thermoregulation, right? So when you are exercising well-controlled room temperature, then people are running outside in very cold conditions and very hot conditions. What do we know, and how are the responses of a stress response protein, whether heat shock protein or cold stress induced proteins?
Right.
What do we know about the interplay of those conditions in terms of SPM levels and resolution of
Yeah,
inflammation?
great question. So regarding heat, to my knowledge, I don't think anyone's actually looked to see how increasing body temperature impacts production of these. But there are several studies looking at cold exposure. So we know with cold exposure, there's activation of brown fat, right? Brown adipose tissue. And that seems to be driven by catecholamines.
Mhm.
And so they have found, actually Dr. Spite has found that cold exposure leads to increased production of SPMs from brown fat. So I'm not aware of the literature on increasing temperature, but it seems to be that decreasing temperature.
So I think that's like something that we can follow up, because especially we have these stress response proteins at various levels, like the transcription factor as well as target protein.
Right.
And then talking about epinephrine, right? So here you are talking about resolution of inflammation and epinephrine. Epinephrine is also a platelet activator,
Right.
right?
Yeah.
So the question I have for you is, how does, like, if you have COX-1 inhibitor like aspirin, right? Then that has beneficial effects. How is like epinephrine having different effects on different cell type versus in your resolution of inflammation?
Yeah. So, I mean, obviously we've only focused on macrophages, but there's a whole body of literature studying other effects of epinephrine signaling, right? Cardiomyocytes, platelets, in the bone marrow.
Add platelet activation markers in those studies, so even to rule out maybe you are not at the level where you are causing platelet activation kind of situation.
Yeah, and kind of tapping in what you're alluding to with the stress response. So what we see with exercise is that there's these bouts of so-called stress, right? And so you kind of think of it as that there's just like an acute exposure to epinephrine. Whereas in these more chronic stress models, epinephrine is staying elevated, and that's probably leading to downregulation of the receptors. So I think there's a lot here that we still don't understand. But I suspect what you're kind of suggesting is going to be dose and duration dependent.
Yes.
And it's obviously going to be cell type dependent. So I don't have a great answer for, that, but yeah, you're right. We should look at, what's happening in platelet markers circulating. We actually haven't done much looking at how this affects circulating immune cells and their production of SPMs. We do see that one of the other SPMs, Merizin-1, goes up in circulation following exercise. So there may even be differential SPMs that get upregulated following exercise. So yeah, there's a lot there that we just don't know.
But you are measuring in the first bio trial.
Yes.
So,
Yes.
so you could point us with that information.
Right.
Speaking of which, jobs here. He's our chief fellow and very research oriented. So extending NYU to Louisville population with cardiology support and getting the fellow involved. That would be the guy.
Okay. Okay, great. Now I've got
Steve Jones has a question. He's
Oh.
online.
Sure, go for it, Steve.
Hey, Jason, great job. I had two questions. One, are there genetic mutations that impact, 1215 or any of these enzymes involved in the production of SPMs? Has that been documented as one? And just because that's kind of more of a yes, no. The second is, well, I'll wait on that. Then the second is a bit more involved.
I should know this. I think there are, but I honestly, off the top of my head, I don't recall specifically what the mutations are.
Yeah, because I would assume it's kind of complicated and it would be difficult to ascribe like one specific gene unless it's say like FPR2, right? To a function. So the more important question was, every time I see it, it still strikes me the magnitude of kinetics and the leukocyte clearance from the peritoneal cavity. So like with the prazosin experiments. So I think you're counting neutrophils, right? And so you see the spike and then the resolution. I mean, I get your point that downward slope is much steeper, but is there room for just a more aggressive, more robust, or maybe more efficient immune response that's going on? I mean, yes, resolution may be part of it, but the fact is, I mean, you see, when you first look at it, what's most striking is the overshoot in the exercise group, right, over the sedentary group, if I'm remembering this correctly.
Yep.
And so I'm wondering, to kind of get to that, is it only so there could be two things going on. One, there's this hyperactive response or hyper responsive phase in terms of the immigration of leukocytes to the peritoneal cavity. And it could be a separate process. They egress or they're disposed of much more quickly in the exercise group. And I'm wondering, could you do an experiment where you have sedentary and exercise and you just inject the neutrophils, so skip the zymosan part, and say take activated neutrophils and do different loads? And I think if you did that, wouldn't you be able to see that, oh no, there's clearly it's a disposal or a clearance, a resolution defect. There may be something else going on, but then you could kind of isolate that and pin it down separately. And then I still think it's super interesting the fact that the response is so much more robust. What are your kind of thoughts about that?
Yeah, no, I think no, those are some of the questions that I'm still kind of bouncing around in my mind too when I see those data. So, I mean, yeah. So to your point, is it really all macrophage driven, right? Or is there some effect on the neutrophils? And it, Marston's data in our group looking at different neutrophil populations. It might be with exercise we're actually recruiting a different neutrophil population with exercise, and those neutrophils, for whatever reason, as they interact with macrophages, produce more SPMs, and then they resolve faster. So I think there's room here, yeah, for a lot of other contributing mechanisms that we haven't just looked at yet. Yeah, absolutely.
Okay, thank you. Yeah, I wasn't even considering that there are different populations, perhaps.
Yeah.
That's interesting. Yeah, maybe with fate mapping studies, you guys could be able to figure out, are those like marginated pools that are differentially coming in and they're demarginating? And yeah, that's interesting.
Right. Yep. Yeah, Dr. Conklin?
Yeah, I had kind of two major questions. One was about the dependence on epinephrine, as opposed to, wouldn't norepinephrine do the same thing? And it's at all sympathetic endings, and epi really only comes from the adrenal,
Right.
and requires a little bit higher level of activity to stimulate.
Right.
So why wouldn't norepi do the same thing? It acts at alpha ones and
it very well might.
Okay.
But when we looked at macrophages in a dish, when we hit those cells with norepi, we didn't see an effect. That doesn't mean that it's not happening in vivo, but at least in vitro, we didn't see a direct effect with norepi on those macrophages in terms of promoting or resolving D1 production. But the other thing that might be happening is that epinephrine's reprogramming circulating monocytes to some extent, and then when they get into the adipose tissue, then they're interacting with some other environmental factors within the adipose tissue to become more M1 or M2. So Yeah, I mean, norepi might be playing a role in the tissue, but yeah, we just didn't see any evidence to suggest that that's occurring. But I wouldn't say that it's not happening.
Yeah, I think that's interesting, and just to reflect on your Perspire trial, that you're doing exhaustive
Right.
exercise, and so I would hope then the measurements of epinephrine in the urine will actually reflect
Right.
that that's a higher level of stimulus, for whatever reason that's required. The second has to go back to the diet dependence.
Mhm.
Because I noticed in the initial studies, probably we're using C57 black 6
And the
mice.
peritonitis study,
Yeah,
well,
with
it's all FEP.
FEB,
Yeah.
okay. But the initial studies were like normal rodent chow, and then I saw with your high fat, you had a low fat diet, which is not
It's not
the same.
Right.
So my question is, rather than looking at the high fat diet for what it has that maybe more pro-inflammatory, the saturated fat, for instance, what in low fat and normal chow is actually allowing this process to happen?
Yeah.
Like,
Yeah.
is there really a diet dependence, or is it just that that particular high fat is inhibitory to the
inhibiting.
process?
Yeah.
Because we're talking about switching up diets. Is there something that we're removing that's beneficial, or are we adding things? Can we be looking to add things that benefit? Like you were saying, omegas
Right.
are considered beneficial, but are they the key feature?
Right. Yeah. That's a tough question. But yeah, it could be that the high fat diet is somehow inhibiting directly. But I mean, given the data directly on the adrenal gland and affecting P and T expression, I do think it's that's not happening with normal chow and low fat. So, what are the beneficial aspects of low fat and normal chow diet? It could be that the balance of omega-3 to omega-6, that are just at the right ratio. So in the high fat, there's more fat in general.
Yeah.
More omega-3 even as well compared to normal chow and low fat. But the ratio is off. So I don't know if that's playing an important role here. Yeah, I don't know, to be honest. Yeah. I can speculate, but
Can I follow that question up with a microbiome question? Because I wouldn't be surprised. So when you change around the diets, often fiber is one of the things that goes, especially in the high fat diets. And I'm curious, I think we're learning more and more about secondary metabolites from microbial communities that may be then affecting human health. Have you looked at, I'd be particularly interested in your exercise versus, on the same diet group,
right.
what shifts you might be having in the microbiome that.
We haven't, others have. They've looked at how the microbiome changes in response to exercise, and there's definitely changes that occur there. There is data, published literature showing that microbiome does seem to affect systemic SPM production. So all of that to say, the strong rationale to look at exactly what you're suggesting, but we haven't. But it's plausible. Yeah.
In the career trying to answer just the questions that we got after that.
All interesting,
Yeah,
questions.
So why we're doing research grant rounds and cross fertilization, and the ideas are really tremendous. All right, well, if no other questions, thank you very much.
Thank you.